Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing CSIR PYQs 2016 and 2017. I will be discussing only the genetic section. If you are new to my channel, please do like, share and subscribe. Coming to the next year, June 2016. Which one of the following statement is incorrect? Quantitative inheritance results in a range of measurable phenotypes for a polygenic trait. That is correct. Polygenic trait often demonstrate continuous variation. That is also correct. Certain alleles of quantitative trait loci have an additive effect on the character or trait. That is also correct. Alleles governing quantitative traits do not segregate and assort independently. That is not correct. So the answer here is option 4. Then next one. A mouse carrying 2 alleles of insulin-like growth factor is normal in size, whereas a mouse that carries 2 mutant alleles lacking the growth factor is a dwarf. The size of a heterozygous mouse carrying 1 normal and 1 mutant allele depends on the parental origin of the wild type allele. So such pattern of inheritance is known as genomic imprinting. In genomic imprinting, the expression of the gene depends on whether they are inherited from the mother or from the father. So here they have told the size of the heterozygous mouse carrying 1 normal and 1 mutant allele depend on the parental origin of the wild type allele. Such a pattern of inheritance is known as genomic imprinting. Next one. Which one of the following statement is incorrect? Loss of genetic variation occurs within a small population due to genetic drift. That is correct. The number of deleterious alleles present in the gene pool of a population is called genetic load. That is also correct. Genetic erosion is a reduction in the level of homozygosity. It is not correct because genetic erosion it leads to drastic or diminishing or complete loss of unique genes. So answer here is option 3. Then next one. Which one of the following statement is correct for amplified fragment length polymorphism? PCR using a combination of random and gene specific primers that is used in AFLP. So that is correct. Option 1 itself is correct. Next one. Mini satellites are used as markers for identifying individuals via DNA fingerprinting, as alleles may differ in the number of repeats. From the southern plot shown below, identify the progeny A, B, C, D for the given parents M mother, F father. So the progeny will have 1 band from the father and 1 band from the mother. So you have to see for that. Now if you take the case of A has got 1 band inherited from the mother as well as 1 band inherited from the father. B also has got 1 band inherited from the father, another band, second band inherited from the mother. C is just the same as the mother, so it is not an offspring, whereas A and B are offspring. Now coming to D, it also has 1 band inherited from the father and the second band inherited from the mother. So D is also the offspring. So A, B and D are offsprings. So answer here is option 2. Then next one. Poplar is a dioecious plant, a wild type plant with the 3 genes capital A, capital A, capital B, capital B, capital C was crossed with a triple recessive mutant small a, small b, small b, small c. The F1 male hybrid capital A, small a, capital B, small b, capital C, small c was then backcrossed with the triple mutant, and the phenotypes recorded as follows. So this is the case of a 3-point test cross. The distance in map units between A to B and B to C. So you have to see for the parental phenotypes. So the parental phenotypes are capital A, small a, capital B, small b, capital C, small c, that is maximum in number, so 300. Then next one will be small a, small b, small b, small c, small c. They are maximum in numbers. So that will be your parental types. The double crossovers. Small a, capital B, small b, small c, and capital A, capital B, small b, capital C, small c. They are your double crossovers. The rest will be your single crossovers. So from the double crossover, you can come to know the gene order. So if you see here, these are the parental, these are the double crossovers. Now which gene is changing its position when compared with the parental? It is B that is changing the position. So B is in the middle. So the gene order is capital small a, B and C. Now single crossover between A and B. So these are the parental. The single crossover takes place here. These are the offsprings. If you see the tabular column, you can come to know how many numbers are there. So 100 and 120. And single crossover between B and C, it is 65 and 75. Now A to B map distance is single crossover between A and B plus double crossover by total progeny into 100. Substitute the values and you will get 25 map units. Similarly, B to C map distance. Single crossover between B and C plus double crossover by total progeny into 100. Substitute the values, you will get 17 map units. So the answer here is 25 and 17 map units. Option A. Then next one. Two interacting genes independently assorting were involved in the same pathway. Absence of either gene's functions leads to absence of the end product of the pathway. A dihybrid cross involving two genes is carried out. What fraction of the F2 progeny will show the presence of the end product? So, capital A, capital B, small b will be your parents. These are the gametes. This is the F1 progeny. F1 on selfing, you will get F2 progeny. This is the dihybrid cross, the Punnett square. And they have told that absence of either genes. If A is absent or if B is absent, leads to absence of the end product of the pathway. So we need to find out what fraction of F2 progeny will show the presence of the end product. So those which have both capital A and capital B in them, in their genotype, will show the presence of end product. So how many are there? 1, 2, 3, 4, 5, 6, 7, 8 and 9. So that will be 9 out of 16. Answer is option 3. The next one. 5 bacterial markers were followed for the co-transduction experiment. The following table documents the observation of this experiment. Plus denotes co-transduction and minus denotes lack thereof. ND stands for not determined. Pick the correct order in which the genes are arranged on the bacterial chromosome. So first we will take arginine into consideration. Arginine goes along with galactose. Then leucine goes along with STR, and STR goes along with both galactose and leucine. So STR goes along with galactose and leucine means STR should be in the middle of galactose and leucine. So first will be arginine, then comes galactose, then STR, then leucine. And methionine, it goes with leucine. So methionine in the end. So the answer here will be option 4, arginine, GAL, STR, leucine and methionine. Answer is option 4. Then next one. Molecular polymorphic markers are already known with respect to tobacco mosaic virus resistance in tobacco. Among these, which marker system will you select that is simple, economic and less time consuming? So out of these markers, which is simple, economic and less time consuming? It is RAPD. Answer is option 1. Then coming to the next year. So December 2016. So first question, if non-disjunction occurs in meiosis 1, which of the following scenario is most likely to occur? So non-disjunction occurs in meiosis 1. So these are the chromosomes. Here is where the non-disjunction is occurring during meiosis 1. They will get separated, the chromatids will get separated in meiosis 2. One of the cell will be normal and this will be aneuploid. So here you will get two gametes, that is both the cells will be aneuploid. Okay. Here you will get gametes n+1, n+1, n-1 and n-1. So two gametes will be n+1 and two will be n-1. So the answer here is option 1. Then next one. Which of the following mutagen is most likely to result in a single amino acid change in gene product? So acridine, orange, ethidium bromide, all these are intercalating agent. They don't cause a single amino acid change. So single amino acid change is brought about by ethyl methane sulfonate or EMS. Then next one. Which of the following is true for cells harboring F' plasmid? So cells containing an F plasmid with some bacterial genes in them, they are called as F' plasmid. And F factor, if it does not excise from the bacterial when an F factor excises from the bacterial chromosome, a small amount of the bacterial chromosome also goes along with the F factor. And these chromosomal genes will then be carried by the F plasmid and it is called as F' plasmid. So their F plasmid is non-functional. That is not correct. They have asked for which of the following is true for cells harboring F' plasmid. They exhibit increased rate of transfer of all chromosomal genes. No. All chromosomal genes are not present in the F' plasmid. They are merodiploids. Answer is option 3. They are merodiploids. Then next one. 3 MET minus E. coli mutant strains were isolated. To study their nature, the mutation, these mutant strains were treated with mutagens, EMS or proflavin, and scored for revertance. The results are obtained is summarized below. Plus stand for revertance and of the original mutant and minus stand for non-revertant obtained. Based on the above, the typical mutagenic effect of EMS and proflavin was what was the nature of the original mutation in each strain. So A is getting reverted by proflavin. So proflavin is an intercalator and it causes frame shift mutation, maybe insertion or deletion mutations. So A is getting reverted by proflavin means A should have been insertion or deletion of a single base. So that is seen in option 3. A, insertion or deletion of a single base. Now B is getting reverted by EMS. EMS cause transition mutations. So B is getting reverted by that means B should have been a transition mutation. So answer is option 3. And C is not getting reverted by those EMS and proflavin. So it should have been deletion of multiple bases. So the answer is option 3. Then next one. The following scheme represents deletions 1 to 4 in the R2 locus of Phage T4 from a common reference point. This is the common reference point. This one is the common reference point. The bars represent the extent of deletion in each case. 4-point mutation A to D are tested against 4 deletions for their ability or inability to give wild type are 2 plus recombinants. The results are summarized below. Based on the above, the predicted order of the point mutation is. So I have redrawn the picture over here. This region of deletion is common to all of them, but here from here onwards there are some variations. So first one, deletion is there in deletion 2, 3 and 4. So you give a minus sign for that, but deletion is not there in the first one. So you give a plus sign for that. Now compare this sequence, that is plus, minus, minus, minus with the table. Where does it come? It comes for D. So the first gene would be D. Then next one. Here plus because both these there is no deletion, whereas deletion occurs in third and fourth, so you give a minus sign. Plus plus minus minus, where does it occur in the table? Plus plus minus minus for this particular deletion 2. So that is with the B gene. Then next one, plus 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 minus, that is seen in case of A. Then plus 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 is C, that is seen in the case of C. So for the tabular column, these 1, 2, 3, 4 represent the deletions. Okay, I forgot to tell that earlier. So based on this tabular column as well as from this figure, you will get the gene order as DBAC. That is your option 2. Then next one. The following pedigree show inheritance pattern of a trait. So this is the pedigree and it shows the inheritance pattern of a particular trait. From the following, select the possible mode of inheritance and the probability that the daughter in generation 3 will show the trait. So this daughter should show the trait. What is the probability for this daughter to show the trait? Now, since the disease is transmitted from father to son, this is the father and the disease is transmitted to the son, it is not any X-linked recessive disease. So out of these options, 1 and 2 can be cancelled. Autosomal recessive is the answer. So this pedigree represents autosomal recessive condition. So these are the genotypes for this particular pedigree. So small a will be the one for affected individual. So wherever affected individuals occur, it is small a. Now since this individual or the son is affected, now the mother should be heterozygous. So based on that, you will get that this offspring is also heterozygous for the disease. Now she got married to a man who is having the disease trait. So capital A small a into small a small a. So capital A small a into small a, these are the gametes, and out of that you will see that the probability that the daughter in generation 3 will show the trait is half. So the answer here is option 3. So coming to the next year, June 2017. A recessive inherited disease is expressed only in individuals of blood type O and not expressed in blood groups AB or AB. Alleles controlling the disease and blood groups are independently inherited. A normal woman with blood group A and her normal husband with blood group B already had one child with the disease. The woman is pregnant for second time. What is the probability that the second child will also have the disease? So it is a recessive inherited disease. It is expressed only in individuals with O blood group, not in those with blood groups A, B or AB. Now a normal woman with blood group A and her normal husband with blood group B has a deceased child. That means the woman is heterozygous for blood group A and husband is also heterozygous for blood group B. Since the child is deceased, that means he or she, the child, is having small o genotype. So in order to get small o genotype, the parents have to be heterozygous. So these are the parents, AO into BO. They are the parents. Now probability of getting O from the mother is half. Probability of getting O gene from the father is also half. Probability that the child is deceased is 1/4. Now probability that second child is deceased is half into 1/4, that is 1/16. So the answer here is option 3, that is 1/16. Then next one. The following is a schematic representation of hypothetical pathway involved in formation of eye color in an insect species. So white pigment converted by product of gene A to vermilion pigment. Similarly, white pigment Y converted by product of gene B to brown pigment. Now mix of both the pigments leads to the red dye color. Genes capital A and B are linked and have a map distance of 10 centimorgan. Females with genotype small a plus, small a, B plus, small b are test crossed. Further, in these females, the two genes are linked in cis. A plus and B plus represent wild type alleles, while A and B represent the null alleles. The progeny of the test cross have individuals with 4 different eye colors. What is the expected ratio of individuals with eye color red, vermilion, brown is to white in the progeny? So this is the hypothetical pathway for the formation of eye color in the insect species. Of these, genes capital A and small capital B are linked and have a map distance of 10 centimorgan. The female genotype is small a plus A and small b plus B and are test crossed, that is crossed with the recessive parent. And the genes in the females, that is A plus and B plus, they are linked and are in cis orientation. So they are on the same side, on the same chromosome. So this is the genotype of the female. It is test crossed with the recessive parent. So A plus, B plus, they are in cis orientation. So they are on the same chromosome and they are 10 centimorgan apart. And these are that of the recessive parent. So these are the gametes. A plus, B plus, AB, A plus, B and A, B plus. That are the recombinants. Into the gametes of this parent will be only one, A, B. Now upon crossing you will get A plus, B plus, AB, A, B, A plus, B and AB, AB plus, AB and AB, AB. So out of these 5%, no, they have told it is 10 centimorgan apart. So they are the recombinants. The recombinants will be 5% each, as they have a map distance of 10 centimorgan or 10% recombination frequency. So 5% each will be your recombinants, that is vermilion and brown. Red and white are the parents. They will be 45% each. So red is to vermilion is to brown is to white will be 9 is the ratio will be 9 is to 1 is to 1 is to 9. So the answer here is option 3. 9 is to 1 is to 1 is to 9. Then next one. A phenotypically normal fruit fly was crossed to an other fly whose phenotype was not recorded. Of the progeny, 3/8 were wild type, 3/8 had ebony body color, 1/8 had vestigial wings and 1/8 had ebony body color and vestigial wings. Ebony body color and Vestigial wings are recessive characters and their genes are located on two different autosomes. Based on this information, which one of the following is the likely genotype of the parent? So a normal fruit fly was crossed with another whose phenotype is not known. So normal fruit fly means both the genes are, that is ebony and vestigial body color and ebony body color and vestigial wings are recessive characters. So if you give E for ebony color and VG for vestigial wing, E plus will be the wild type, VG plus will be the wild type. So a phenotypically normal fruit fly was crossed with another fruit fly whose phenotype was not recorded. So your first parent should be your normal fruit fly, that is the one with carrying the wild type genes. So if you see option 1, the first parent is fully recessive, so that cannot be the parent, cannot be the answer. Then in the second case it is only ebony color, smally are present, so that cannot be the parent. In the second case it is vestigial wings, sorry, in the third case it is the vestigial wings which is recessive character, so that also cannot be the first parent. But if you see the fourth option, E plus E, VG plus and VG, so E plus and VG plus both are normal characters and they are dominant over ebony and vestigial wings respectively. So that can be the first parent and this will be the phenotype of the second parent. So the answer here is option. Now next one. In normal individuals, there are 3 MST restriction sites, 2 flanking the beta globin gene and 1 within the gene. In individuals affected by a disease, a single nucleotide polymorphism in the beta globin gene abolishes the internal MST2 recognition site. The RFLP pattern for this locus obtained by hybridization using a probe internal to the flanking MST2 sites from 3 siblings of a family is shown below. So this is the normal son, this is the normal daughter with 2 bands and affected son is having only 1 single band. Okay, based on the above profile, what is the nature of the genetic disorder? Now in normal individuals, there are 3 MST2 restriction sites, that is over here 1, 2, 3, and 2 flanking the beta globin gene and 1 within the gene. So 2 are flanking the beta globin gene and 1 is within the beta globin gene. Now a single nucleotide polymorphism in the beta globin gene abolishes the internal MST2 recognition site. So if you get a single nucleotide polymorphism over here, this MST2 recognition site is abolished from the beta globin gene. The RFLP pattern for this locus obtained by hybridization using a probe internal to the flanking MST2 sites, the 3 siblings of a family is shown below. That is, the probe will be somewhere over here. So based on the above profile, what is the nature of the genetic disorder? Now here the normal son is heterozygous because he is having the normal 2 normal bands similar to that of the normal daughter, and this band, that is 1.35 kb, is similar to that of the affected son. So the normal son is a heterozygote and has 1 diseased band along with the normal band. So it is the case of an autosomal recessive condition. If it was X-linked recessive condition, then the son would be the affected, this is the affected son. Affected son will be having only 1 single band, but the unaffected son, that is XAY, unaffected son will be having normal band, he will not have the heterozygous condition. So it is the case of autosomal recessive condition. This is the normal daughter and this is the affected son. If it was autosomal recessive, then you will get a heterozygous son and an homozygous son. That is why affected son got only 1 single band, whereas heterozygous son got 2 bands. So it is the case of autosomal recessive condition. Answer is option 3. Now next one. In a transduction experiment using A plus B plus C plus genotype as a donor and A minus B minus C minus as the recipient, A plus transductants were selected and screened for B and C. The data obtained are shown below. So A plus B minus C minus is 573, A plus B plus C minus 98, A plus B minus C plus 11, A plus B plus C plus 68. The co-transaction frequencies for A plus B plus and B plus C plus respectively are. Now for A plus B plus. So A plus B plus occurs here. That is at number of recombinants 98, as well as A plus B plus can be seen here, that is 68. So 98 plus 68 divided by this total. Whole total will be 750 into 100. So that will come around 22%. Now for B plus C, you can see it is present only in the 68, number of recombinants coming as 68. So 68 by 750 into 100, so 9%. So the answer here will be 22% and 9%, option 2. The next one. In a breeding experiment, two homozygous parental lines P1 and P2 were crossed to produce F1 hybrids. Due to an experimental error, seeds of these hybrids got mixed up with the seeds of two other germplasm lines P3 and P4 and hybrid seeds derived from them. A marker-based fingerprinting exercise was performed using 6 randomly selected seeds F1 to F6 and the mixed material and the 4 parental lines. Results of this analysis are shown below. Based on the above data, which one of the following options represent the correct set of parents and their F1 progeny? So a breeding experiment was carried out and two homozygous parental lines, that is P1 and P2, were crossed to produce F1 hybrid. Now due to experimental error, what happened? The seeds of these hybrid got mixed up with P3 and P4 as well as their hybrid seeds derived from P3 and P4. So they conducted a marker-based fingerprinting exercise and performed using 6 randomly selected seeds, that is F1 to F6 from the mixed material and the 4 parental lines. Results of this analysis are shown below. So based on the above data, which of the following options represent the correct set of parents and their F1 progeny? So P1 into P2 is F3. These are the P1, P2 is F3. Now you have to remember that whatever genes are present in the bands are present in the parents will be present in the offspring or the progeny also. Now P1 into P2 is F3. So P1 has got a band over here. That band is not present in F3. Okay. P2 band is present in F3. This band is not present. This band is also not present. These two bands are not present in F3. So it is not the offspring. P1 and P2 offspring is not F3. So option 1 is not correct. Now P3 and P4 is F2. P4 one band is present over here. Other bands, these two bands are not present in F2. So P3 and P4 progeny cannot be F2. Then P1 and P2 is F1. So this is the P1 band, yes, that is present in F1. P2 band is also present in F1. These two bands are also present in F1. This band is present in F1. This band is also present in F1. So P1, P2 progeny is F1 is right answer. So the answer here is option 3. So coming to December 2017, 2 mutant plants both bearing white flowers were crossed. All F1 plants had red flowers. When the F1 plant was selfed, it produced progeny with either red or white flowered colors in the 9:7 ratio. Based on this information, which one of the following conclusion is correct? So this question is repeatedly asked for 3 or 4 times, I think. The same question based on complementary ratio. and the same and I have the same and in one of the question the flower colors were different, else the answer was the same thing. Okay, so I have given the explanation for complementary ratio over there. You can refer to my previous videos for explanation. So here the answer will be the mutation in the parents are non-allelic. Answer is option 3. Option 1 sorry, the mutation in the parents do not complement each other, they complement each other. That is why you got red colored flowers upon crossing two white flowers. The mutation in the parents are allelic, they are non-allelic, not allelic. The mutation in the parents are linked, that is also not correct. Then next one, in a linkage map, two genes A and B are 70 centimorgan apart. If individuals heterozygous for both the genes are test crossed, number of progeny with the parental phenotype will be equal to the number of progeny with recombinant phenotype, because maximum recombination frequency is almost 50 centimorgan. So here it is greater than 50 centimorgan. So equal number of progeny with recombinant phenotype. Answer is option 1. Then, a researcher exposed Drosophila larvae to 37 degrees Celsius. During their growth, one of the adult flies that emerged had cross-veinless phenotype. Cross-veinless is a non-mutant in Drosophila. When this cross-veinless fly was crossed to a non-cross-veinless mutant fly, all the progeny had normal phenotype. The observed phenotype can be best explained as an example of phenocopy. Answer is option 2. So a phenocopy, it is a variation in the phenotype which is caused by environmental condition and in such that the organism phenotype matches with that of the phenotype which is determined by genetic factors, not environmental factors. So here the larvae showed cross-veinless phenotype when exposed to 37 degrees Celsius. This phenotype arose as a result of 37 degrees Celsius exposure and not a genetic cross. That is why you got all normal phenotype when crossed with an original cross-veinless fly. Answer here is option 2. Phages are collected from an infected E. coli donor strain of genotype cysteine plus, leucine plus, threonine plus, and used to transduce a recipient of genotype cysteine minus, leucine minus, threonine minus. The treated recipient population is plated on a minimal medium supplemented with leucine and threonine. Many colonies grew. Which one of the following combination of genotypes are appropriate for the colonies that grew? So cysteine plus, leucine plus, threonine plus, cysteine plus, leucine minus, threonine plus, cysteine minus, leucine plus, threonine minus. Cysteine plus comes, cysteine minus comes over here, so it is not correct. The treated, because they have told that the treated recipients were plated on minimal medium supplemented with leucine and threonine. So those which are leucine minus and threonine minus can also grow in the minimal medium. Naturally, those with cysteine plus will 100% grow in the medium. But cysteine minus cannot grow in the medium because cysteine is not provided to the minimal medium. But the first gene to enter will be normally cysteine. So cysteine would have already entered. So which one of the following combination of genotypes are appropriate for the colonies that grew? So cysteine minus, those colonies with cysteine minus will not grow. So options wherever cysteine minus is present, you can consider it as not correct. So option 2 also cysteine minus is present. Option 3 all are cysteine minus. So that is also not correct. So option 4 all are cysteine plus. And you can see that leucine minus, threonine minus, they will grow. Cysteine plus leucine minus threonine plus, that also will grow. Cysteine plus leucine plus threonine minus, that also will grow. So the answer here is option 4. Then next one. The following table shows mapping data for 3 interrupted mating experiments using 3 different HFR strains and an F- strain. Appearance of gene in F- cells. These are the HFR 1, 2 and 3, and these are the time. The time is represented in minutes. The following answer are derived. The order of genes is, the distance between. The correct combination of answer is. So here if you see, B, D, C and F. B, D, C, F. Again also B, D, C, F. Here also D, C, F. All these are always going together. So B, D, C, F is one sequence. Now coming to E is there, G is there. We don't know which is going first, whether E is going or G is going. So for HFR1, distance between E and F, this distance between E and F is 18 seconds. HFR2, distance between F and G is 32 seconds. And in HFR3, distance between E and F is 18 seconds. So 18 is less than 32 seconds. So after F, E will go followed by G. So E, G, F, B, D, C, F, E, G is the correct sequence, and that matches with option A. So not option A, statement A. So E, G, B, D, C, F is what is the correct sequence. Now coming to the next one, the distance between F and G is 32 minutes. F and G over here, F and G, I already told it is 32 minutes. And distance between F and B is 30 minutes. That is also correct. Then C and E is 28 minutes. Distance between C and this one, C and E is 28 minutes, and distance between B and C is 20 minutes. That is also correct. So the answer here is option 1, that is A, C and D. D is also correct. So answer is option 1, A, C and D. Then next one. The inheritance of a given disorder is recorded in 3 similar families shown below. Based on the above limited information, which of the following inheritance pattern best explains the observation? So both male and female are affected. If you see the pedigree, you will see that both male and female are affected. So even if parents are not deceased, offsprings are deceased in family 1. If it is dominant, then family 3, the offsprings would have got affected, but that is not the case in case of family 3. Now if it was X-linked recessive, then father would have been affected in family 2. But that is not the case. So it is a simple example of autosomal recessive inheritance. Answer is option 3. The next one. In Drosophila, bar I is a dominant mutation, while miniature wings and yellow body color are recessive mutation. Heterozygous females for these mutations were crossed to normal-eyed, miniature winged and yellow body colored males. Assume the following progeny was obtained. So these are the progenies that are obtained. Based on the above results obtained, the order of genes will be, the genetic distance between BY will be, the correct combination of the answer is. So these are the phenotypes and these are the number obtained. Out of these, the one with maximum number, showing maximum number, will be your parentals. So here B, M plus Y plus and B plus My are the parentals. Now double crossovers, if you see, BMY 20, then BMY plus 25, and B plus M plus Y plus 30. These are the double crossovers because they are the ones which show least number, the very less number. So now there are 3 double crossovers. So with the 3 double crossovers, determining the gene order is not that easy. So now if we take BMY as the order, then if it undergoes double crossover, then you will get progeny as. So these are the parentals. The parentals, if they undergo double crossover, you will get BMY plus and B plus M plus Y, which is 45. But B plus M plus Y is not present in the double crossovers over here. So this cannot be the gene order. Now again, if you take the gene order as MBY, if we take the gene order as MBY, so the parentals, this upon crossing, you will get M plus B plus Y plus and MBY, which are your double crossover. These are there, these over here. If you see, these are present. This double crossover sequence is present in this one also, which we have taken. So MBY is the gene order. Now single crossover between B and Y, you will get these progeny for recombination frequency. You can go for number of single crossovers plus number of double crossover by total number of progeny into 100. Substitute the values, you will get it as 40 centimorgan. So the answer here is B and C. Answer is option 2.